thank you so much for presenting this year and being patient with figuring out all of the logistics of making this a virtual experience. You're all have done a really good job turning things in on time, and I really appreciate it.
Okay, everyone, we're going to go ahead and get started since we're on a schedule this morning. So good morning and welcome to the 31st Annual Elkin Isaac Research Symposium at Albion College. Thank you all so much for being here this morning. My name is Elizabeth Palmer and I'm moderating the session along with Dr. Matthew Schoen. Things are obviously a little bit different this year, so this is our live Q&A for our poster sessions, and the posters are going to be presented in front of you on your screens. So each poster presentation will have 5 minutes of Q&A, and each session today is being recorded for documentation purposes. So without further ado, our first presenters today are Samantha Emerson, Aaron Lewis, Kirk Myers, Danielle Reinman, and Liam Smith. And we do ask that you all keep yourselves muted just to avoid feedback, and you can answer all or post all of your questions in the chat. So group one, you can go ahead and feel free to explain your project a bit while we wait for any questions to come in.
Thank you so much for the introduction. Just while you guys are looking over our poster, I guess really quick, I can run through kind of our main purpose and our hypothesis to get this going before any questions pop up. So the main purpose of our study was to assess the sleep quality, sleep duration, and mental health of college athletes. And we did hypothesize that college athletes with higher sleep quality would have higher mental health scores than those with poorer sleep quality. Additionally, we thought that college athletes with longer sleep duration would have higher mental health scores than those with shorter sleep. And we kind of, based off of that, we kind of concluded that sleep quality and duration do have an impact on college athletes' mental health. And to tie it all together, we kind of figure that indicates the importance of appropriate and proper sleep in college athletes because of the repercussions of poor sleep habits and what that can do to one's mental health. So that's kind of what I have for an introduction. Feel free to ask any questions. We all are here to answer anything. So thank you guys for being here.
So the first question for the group here, was this Albion College students only, or did you explore other groups of college athletes?
Yeah, so it was just Albion College student athletes only. The Google Form survey that we sent out to all faculty and staff, but we ended up limiting our sample size to just college athletes here at Albion.
Thank you. So the second question, do you have suggestions to help athletes improve their sleep quality?
Yes. So in prior research, it was found that routines and like establishing good sleep like behavior and manners was found to be like very effective.
Thank you. And actually, the next question here is a bit of a follow-up to that. Actually, based on the results of your research, what would you suggest to other students in terms of sleep health?
To kind of go off what Liam said, to establish habits before going to sleep, like trying to stay off your phone, like probably 30 minutes to an hour before going to sleep, and then also trying to wind down prior to sleep. Some people have found it helpful, and research has shown writing down stressors prior to going to sleep so you don't go to sleep with those stressors will help sleep quality. And just overall establishing proper habits prior to going to sleep will help improve quality and duration.
Thank you. And then I actually have a question of my own. So from what I'm seeing here, mental health is the dependent variable of your study. How did you measure mental health? What was the creation of that? Was that a scale, or how did you choose to measure that?
Yeah, so our survey was kind of compiled from a lot of other like already established surveys. So we used the DASS, which is the Depression, Anxiety, and Stress Scale, and that measured mental health in students. So it kind of asked questions like, do you feel anxious? Like how often? Like it was like, I think like most likely applies to me versus like doesn't apply to me, those kind of like questions. So that's how we were able to, I guess, add a numerical value to people's mental health.
Thank you. One more quick question here. Following your study, did you come up with recommendations for amounts of sleep? Is it different for athletes and non-athletes?
For the amounts of sleep, we actually did have part of our questionnaire filled out specifically on the duration that they were getting originally. And so we were able to look at different groups depending on if they had less than 6 hours, between 6 and 7 hours, between 7 and 8, and then over 9, which was actually really beneficial for us because that allowed us to find the significance between the groups and see where the differences actually laid between the mental health and the sleep quality or sleep duration. So we didn't look specifically at non-athletes, but we were able to look at the athletes and found that there was a significant difference.
Thank you so much.
Thank you so much for all of those questions. That's actually time on the first presentation. So our next one is Christina Rayfell. And just a reminder to keep yourselves muted and keep putting those questions in the chat. So Christina, you can go ahead and get started.
All right, good morning. Thank you for joining my poster presentation today. My name is Christina Rayfell and I'm a senior here at Albion. The purpose of this study was to examine the association between physical activity, GPA, and sleep in college students. It was hypothesized that the more physically active students would have a higher GPA. Additionally, higher levels of physical activity would be associated with a better quality of sleep. And lastly, students who performed vigorous physical activity would sleep more than students who had a moderate intensity physical activity. I found no statistical difference between GPA and intensity of physical activity or sleep and intensity of physical activity. And although this study does not support that conclusion, other studies have still emphasized the importance of physical activity on overall health and on overall sleep and cognitive health. So I will now be taking any questions you guys have.
So first question for the presenter, what research do you feel needs to be done next in this area?
So I just think maybe like a bigger sample size or like collecting data in a different way that's not like survey based. I think if we would have like measured the participants like and had them in a lab, we might have gotten different results.
Thank you. Second question for you. Did you limit this to only Albion College students, or did you go beyond the college population?
It was just Albion College students.
Okay, follow-up question for you. Why do you think your research wasn't significant in comparison to the previous studies that you had cited?
Like I said before, like I think participant bias in like the student reported surveys, and also the majority of the participants were females, and students may have selected to take this survey because they were like doing well academically or were more physically active.
Okay, thank you. Next question for you. Did you look at any correlations between areas of major? So for example, science, non-science, and GPA and sleep patterns?
Can you repeat that again?
Yeah, sorry, I may have tripped over that. Did you check to see if there was a correlation between the major that the student had? So, for example, whether they were majoring in natural science or in something in the non-sciences, and whether that affected their GPA and sleep patterns?
We didn't look at the majors, so I don't have any correlation on that.
Okay, thank you. And then the next question for you, how are you measuring sleep quality? I'm actually very interested in this myself.
So I used the Pittsburgh Sleep Quality Index, the PSQI, which measured sleep. So it was just like it's already a questionnaire that was developed. So a question that could have been asked to participants would be like, during the past month, how many hours of actual sleep did you get at night? Which may be different than the number of hours you spent in bed. And then the participants typed in a numerical amount for the number of hours of sleep that they had obtained per night.
Thank you. Any further questions?
Thank you.
Thank you.
And if we're done with questions, it is about time to move on to the next session. So thank you, Christina. So our next presentation is Austin Begley, Jacqueline Espinosa, Allison Haskell, and Nicole Sweetland. And just keep yourselves muted and keep putting questions in the chat. And you guys can go ahead with your presentation.
Hi. So we looked into the effects of physical activity and sleep quality on college students and faculty. So pretty much anyone at Albion College was open to take our survey-based questionnaire. And when we looked into the effects, we kind of also wanted to dive into like the different kinds of physical activity, like moderate and vigorous, as well as no physical activity, on how well students slept, if they slept better or slept worse. We hypothesized that vigorous activity would make you sleep better at night. We figured this because you're tiring yourself out more. You should be more tired and making you sleep better at night. But we actually found nothing significant about our data. So yeah, so I'm open to any questions.
Questions for the presenters? First question for you, is there too much exercise that would poorly impact sleep?
So our study didn't really look at too much exercise. We're actually just trying to figure out, how much exercise would have a significant impact on sleep or whatnot. I think something that you can think about is if you do work out probably too much, and I think too much is kind of a vague number. I think, the physical activity guideline says you want 150 minutes a week of moderate aerobic physical activity or 75 minutes a week of vigorous aerobic physical activity. I think any more than that could be qualified as too much. It just depends, I think, on who you are as a person. And that probably would affect your sleep quality just by, maybe you sleep too much possibly. Or we didn't do any work on that, but that's just something to think about, I think.
Thank you. Next question for you. Did you look or ask your did you ask your respondents the time of the physical activity? So meaning the time of the day in which it was performed?
No, but I think that would have helped a lot. And I also think like during non-COVID times, like a non-survey based experiment like would have been best for our study. So I feel like there was like a lot of limitations like that. And I feel like time definitely could have had an impact, whether it was morning or night. And I feel like also what you ate could have had an impact like for physical activity and how you sleep at night as well. I feel like a lot of things could be added into this study if like we could have in-person participants.
Yeah, certainly. So the next question for you from Dr. Webb, and this is of personal interest to me as well. Did you compare quality and quantity of sleep between professors and students? And if so, were there any observations related to those differences between professors and students?
I can take it. We actually surprisingly, we did not have that many participants that were outside of students. I think it would be very interesting to look at it since they both have like way different lifestyles, way different stressors and everything else, and different like time limits to be able to have physical activity. But like I said, sadly, we didn't have enough faculty to be able to do that in comparison. But I feel like in the future, it would be something very important to look at.
Yeah, sure. Sample size really dictates a lot of what you do. Okay, next question for you. How did you differentiate between vigorous and less vigorous sleep? Or perhaps that's meant to mean exercise. I'm not sure.
So there wasn't really a difference, like a definition difference in the survey. And that was part of our problem that we realized was we don't think people know exactly what moderate and vigorous exercise means. So some of our numbers were like way off and we're like, there's no way this person could be exercising 150 minutes of vigorously intensity exercise in a day. So that was part of our problem with the survey.
Okay, thank you. And then the next question for you, would you like to revisit this research post-pandemic? This was touched upon a little bit earlier, but how would you maybe do this a little differently without the pandemic restrictions?
I think possibly we would just be looking at in-person participants and maybe comparing like one person to vigorous and one person to moderate, like on a span of multiple people. And like I said before, like looking at the time, looking at like if their faculty or students is important. And I also feel like diet is also really important in this. So I feel like a lot of things like could have been looked in deeper. We had a lot of limitations for surveys, like overestimating how much you exercise for the survey or overestimating like how little sleep you got at night just because you feel tired. So I feel like a non-survey base definitely would have been best for this study, but for the time being, that's what we could do for the times.
Certainly. Thank you.
And thanks everyone for those questions. That was really great, great presenting. We are going to take a 5-ish minute break and get started with our next presenter around 9:50. Okay, everybody, I know that wasn't quite 5 minutes, but in the interest of time and just staying on schedule, we are going to go ahead and get started with our next presenter. So Luis Angel Ramirez Galvin, if you'd like to go ahead and explain your project. And just as a reminder, everyone, please keep yourselves muted and post all of your questions in the chat. Thank you.
Hello, can you guys hear me? Can you guys hear me?
Loud and clear.
Okay, good. Thank you. Well, good morning, everyone. My name is Luis Angel Ramirez, and I'm a senior here at Galvin College. I interview community program educators to understand the effects of the COVID-19 pandemic on their youth programs. Just as schools, community programs are a resource of education. And they continue to work through this pandemic. I think it's important to know what they're doing in this time, and also do our best to help them out, just as we would do for any other educational resources that we have in our community. So yeah.
So we have a question for you. This summer, there will be similar COVID restrictions, unfortunately. How will this impact the TEN program this summer and beyond?
Okay, so one of the major sources of funding for the teenagers exploring nature at the White House Nature Center is through the Alvin Community Foundation. So like not knowing where those funds are standing in the next year and stuff like that, it's gonna make it difficult for the program to provide what it used to provide. I mean, we would provide meals for kids and stuff like that aside from the programming. So, I mean, all of these programs in the community are not just in need of funds and stuff like that, but all these resources are have to be allocated into all these programs. So not knowing how much funding is gonna be for the 10 program, it's difficult to plan around that. Also the college and the guidelines, just as last summer, it was like the rule of no visitors and stuff like that. So program participants were considered visitors, so they were not allowed to come into campus. And I mean, knowing the case around the nation where other universities and stuff like that are now aiming to continue classes only if students come vaccinated and stuff like that. So not knowing if the college is gonna have all these guidelines about how kids can better be suited for the classroom, or if the program is gonna move outside and stuff like that. It also depends on the student. So this is like a student-driven program. Every student proposes their own program. So knowing what they want to teach and the atmosphere they want to get, based on that, like it's difficult to plan a program when we don't even know the guidelines that are going to happen in the fall and stuff like that. And since it deals with kids, I mean, from the IRB and stuff like that, you have to create a safe environment for all these kids. But yeah, did that answer the question?
Yeah, thank you. And that actually raises the next question. In your opinion, and based on your research, how can remote learning programs on nature be successful?
Yeah, so there's many respondents brought the issue that, I mean, these youth education programs are more about interaction and creating that bonding within the students and stuff like that. So not having that mutual interaction with each other. And I'm not talking like the 6 feet apart and stuff like that, but rather like seeing themselves present in the environment that they see. I mean, talking to one of the respondents, they had to create a whole new module to create like an interactive walk in nature and stuff like that. And I mean, you don't get things from that you otherwise would get when you're present. But what's it called? Yeah, that's it.
So then following up on that, are there any innovations that you think would really benefit youth programs in a post-COVID restriction world?
Yeah, so just as many programs in Albion, they had to continue their programming through the summer. I know we like to think that, through the summer, schools closed, so we had some relief, like creating these safe environments and stuff like that. But the reality is that these programs continue through the summer. And something that I learned is that they use the outdoors, and the hybrid module of online and in person, and bringing both of the best worlds, I guess, like brought their participants up, yeah, that's it.
Thank you.
And thank you so much. Your time is up for this session. Thank you for those questions. Our next presenter is Angela Myers. So you can go ahead, Angela.
Hi, I'm Angela Myers. I'm a senior biology major and Spanish minor here at Albion. And for my project, I reviewed 107 recent mate choice studies from 2006 to 2020 to see if there was still a taxonomic and sensory bias that was previously identified in a paper published in 2009. And I found that in recent literature, there's been an increase in mate choice studies on spiders, as well as an increase in chemical and multimodal mate choice studies.
So first question for you, why so few mammal studies recently?
I'm not sure entirely because communication is very widespread throughout the animal kingdom. So that's why Coleman kind of pointed out, like, why are there not many studies on it? So mammals definitely use it. I would assume I would think mammals might be harder to study in the lab, whereas like other taxonomic groups that are smaller, like insects and frogs, you can kind of keep them in a lab better and breed them. They have like short generation time, so a part of it might just be the ease of study.
Thank you. Further questions? So next question for you. Were there studies looking at different life stages? So for example, larvae versus adults, and did they show differences between those life stages?
No, I believe that all the studies I looked at only looked at adult animals and the preferences that they use to assess mates.
Okay, thank you. And then the next question for you, looking at your research, what was your favorite animal to study? And I guess why?
The animal that was studied most specifically in the multimodal studies were spiders, and a lot of them are specifically looking at wolf spiders. And I think they're really, really fun and interesting because the males, to attract females, they do like a leg tapping display, but they also simultaneously, that tapping produces vibrations through the substrate. And it's kind of interesting because even if they're like in a dark environment or if they're like burrowed underground, they can still maintain that behavior. They can, instead of the visual display, they can switch to the vibrations to still attract mates in that different environment. And a fun fact is a lot of the wolf spider studies in the recent literature are being conducted by Aline Hibetz and George Utz, both of whom are Albion alums. So there's like a really cool connection there.
Very good. Thank you. I believe we can get one more question in. Are there any taxonomic groups that you believe should be studied more based on perceived importance by researchers?
There was not. I feel like there should be more studies on invertebrates, specifically crustaceans. This wasn't part of this study, but for another class, I read a paper about how pile driving, which is like when bridges, like columns are like hammered into the bottom of lakes or rivers or oceans to build bridges and other structures. It actually, even though that was noise in the acoustics channel, it actually altered hermit crabs' ability to use chemical reception to locate nearby food sources. Yeah, so the human interference is having a very complex impact on animal communication, even in ways that we may not have thought about before.
Okay, thank you. So that time's up, but I want to read a quick comment from Dr. Cahill. Need more snails. Great work with a large topic. So
Thank you, Angela, and thank you for all of those great questions. Our next presenter is Jessica Garcia Lopez. And Jessica, you can go ahead with your presentation.
Hello, I'm Jessica Garcia Lopez. I'm a junior in Albion College and a bio major. I did my presentation on microinvertebrates in the Kalamazoo River. Specifically, we were targeting the wild rice and other areas surrounding the wild rice. And we found that the most diverse area is the wild rice, which was pretty interesting.
questions for the presenter? Okay, first question for you. How did you determine the diversity?
We determined the diversity by counting. I use an identification ID index, and by that I was able to categorize these microinvertebrates based on their taxonomy, and from that I used calculation to find the diversity.
Okay, thank you. And why do you think, based on your research, why do you think the wild rice area was the most diverse?
I think this research is going to continue during the summer, but right now, one of the conclusions we're heading towards is that the wild rice provides a very unique habitat and for the area, and also provides a nesting and resting place for birds. So that could be a factor of why it's so diverse. And in a further investigation, we're gonna try to investigate if there's any changes in between the seasons to see if there's like any diversity differences between the seasons.
Okay, thank you. Next question for you. Do low oxygen levels hurt bioindex?
That's a good question. That's something we're trying to figure out using some further instruments in the Kalamazoo River. But right now we just focus on the microinvertebrates that we collected in the Kalamazoo River.
Okay, and a follow up to that, did you find anything besides invertebrates? Were there surprises in there?
Yes, we found a lot of terrestrial invertebrates in the wild rice. which was pretty interesting because there was nothing except for the beds of the wild rice, which was pretty interesting because it was cold, it was fall. But we hope to see if there's any changes in the summer.
Okay, and then the next question for you, can you discuss the physical environment of wild rice a little bit? So for example, still or fast moving water, sediment type, shade versus no shade. So yeah, just some background on the physical environment.
The wild rice has a very, it grows in a very not so fast flowing river, a very calming. So that's why the Kalamazoo is a perfect place for the wild rice. And also it has, like I said, it has dense stops which provides rooting and floating for the area. And yeah, and it's a place for many nestings for bird species.
Okay, thank you. And I believe one more question we can get in. Pardon me. What changes in microhabitat does wild rice facilitate to make favorable microhabitat? Can you compare to other grasses or sedges in the area?
That's where my further investigation is going to play. I'm going to sample a freshwater marsh in California and see if there's any differences in microinvertebrates and see how urbanization affects microinvertebrate species, because the freshwater in California, it's surrounded by very urbanized areas and buildings, and it's by a freeway which releases a lot of carbon. So yeah, we're looking more into that.
Thank you so much.
Great. Thank you so much for those great questions. Thank you, Jessica. And our next group to present is Bree Ruger, Bach Tran, Rosemary Hernandez, and Sage Joyce. And just another reminder to keep putting those questions in the chat and to stay muted for anyone who's new and just joining us. And you guys can go ahead and get started.
All right, I can start us off for my group. I'm Bree Ruger. I'm a sophomore bio major here at Albion, and our project was looking at the macroinvertebrate species diversity in the Maple River salt marsh. We were specifically comparing between a freshwater area near the salt marsh and the actual marsh, and they're in the same floodplain, but they have different salinity levels. So we were looking at the differences in the community compositions of the macroinvertebrates between the two, and we ended up finding that the community composition between the two were different from each other.
So the first question for the group, any salt endemics?
I actually don't know the answer to this question. If anybody else in the group knows, they can shoot for it, but I am not sure.
Okay, Dr. Cahill says it's complicated in the chat. I will take her word for it, absolutely. Next question for the group, what is a salt marsh?
So the salt marsh is where the one in Michigan is where there was an ocean over Michigan prior, and then it left behind natural salt deposits in the ground. So this specific area in Michigan, there's salt deposits in it, and it makes the salinity higher in that area than the surrounding areas.
Okay, so you mentioned there once was an ocean over Michigan. How long ago was that? Are you familiar with the geological time span there?
I'm not sure of the exact date, but yeah, a very long time ago.
Okay. What's next? Any guesses for why the seeps don't appear to cluster on the NMDS?
So on the NMDS, so on the left side, we have the west freshwater location, and then on the right, we had the steep location. So we're thinking that because of the salinity difference, it causes different communities to comprise in each of those locations. So they created different communities based on their salinities.
Okay, thank you. Next question for you. Sample collection began in 2018 and continued in 2019. Has anything happened since, now that we're hearing this in 2021?
No, we have not been able to go out and collect anything else because of the pandemic. So we have been working with a lot of the data that we got in fall of 2019 and just seeing what other patterns we can find in that data.
Okay, thank you. And then I see a follow-up in the chat. 400 million years ago for that ocean, which coincidentally is how old I feel when I get out of bed in the morning. Okay, one other question for you. Besides salinity, are there any other variables that would affect biodiversity in these sites?
So we only looked at specifically the salinity when we went out to the sites, but there definitely could be other factors that are affecting that biodiversity. When we went out there, we would just measure the salinity and then take samples, but there could be other things that are affecting the community compositions.
Okay, thank you.
Okay, and that's just about time's up. So thank you all for those questions and thank you presenters. Our next group is Alexis Moss, Veda Reddy, and Noelle Robert. And you all can go ahead.
Hello, my name is Veda. I'm Noelle. And I'm Alexis Moss, and we're all biochemistry majors and first years. And our project is on the synthesis of hemidioindigo compounds as potential microtubule formation inhibitors. Millions of people lose their lives to cancer each year. Cancer is the uncontrollable growth of cells and can happen all over the body. To date, one of the most common treatments is chemotherapy, which attacks cancerous and normal body cells. However, our drug, EVL indigo, is derived from HITube-4, which is an alternative cancer drug that inhibits the cancerous cell's life cycle, causing it to die. Our drug has the ability to turn on and off like a light switch, allowing it to selectively only kill cancer cells. This reduces the side effects of cancer treatment on the rest of the body. Thank you all for watching our poster presentation. Would anyone like to start us off with any questions?
Questions for the presenters, please. And please put those in the chat. Okay, first question for you. How does it work to turn on and turn off?
So based on the molecular structure, as you can see in the figure with the light switch in the middle column there, it has that photoswitchability where its molecular structure is able to absorb at a certain wavelength and be able to switch isomers at that double bond there. And the on is like when it's active, and that's in the Z isomer, and that's where it's able to attack the cancer, while off switch is the E isomer, and that's where it won't attack the cancer. And that's the form that the patient would take it in when it's off.
Okay, thank you. Next question for you. What is the difference between your compound and AZO drugs?
So our compound and azo drugs are rely on different wavelengths. So our drug runs at a higher wavelength versus an azo compound, which runs on a lower wavelength, which is 300 nanometers, and EVL indigo runs at 360 nanometers. And adding on to that, because it's at a longer wavelength, that means that it stays in the tissue and stays in effect much longer than azo compounds. So yes, they both have photoswitchability. However, they can attack the cancer much longer than azo compounds can.
Okay, thank you. Next question for you is, how many other researchers are exploring this? Is this a very novel approach, or is this a popular approach within the field of drug creation?
Photosearchability compounds, they are, I would say that they're pretty recent in terms of like the cancer treatment field, and there are a couple other researchers doing it, because like we said previously, our drug is based on HI2/4, which was already derived for, but we just modified it a little bit. So there are other researchers researching this.
Okay, and so then where do you feel that this research should be going next? What's the next question to answer?
So for the future work, we are gonna try to see what we can add on to our drug that we already did. And you can see in the under molecular modeling, the left image, there's a little pocket there. And so we found out that there's room to add stuff onto it to make it more effective. And so we'll do a whole bunch of Suzuki reactions to see how much more effective can we make the drug.
Okay, thank you. And then one more question for you. What is the key difference between your drug and chemotherapy?
So basically with chemotherapy drugs, they work great and it's been successful for many people, but sometimes there are limitations with it. And the problem with it is that it attacks cancerous cells, which is what we want it to do, but it also attacks other bodily cells. So with our drug, what we're trying to prevent is that limitation. We want to target specifically cancerous cells and not like your own healthy body cells. So what we do with it is the patient takes the drug and then we activate it with UV light.
Thank you.
And that is just about time. So thank you so much, ladies. That was great. Our next and last presenter for the poster session is Samantha Dye. And just for anyone who's new joining, just remember to keep yourself muted and to put all of your questions in the chat. And Samantha, you can go ahead.
Hi, thank you. So just a little introduction. Some of this was touched by the last group, but cancer is caused by errors, essentially, that occur throughout the body. One specific type of error found in the hedgehog pathway, which I have illustrated in figure 1, can actually cause many different types of cancers. And so chemotherapy is often used to treat this, but this process can be very harsh on the body because the drug can't tell the difference between the cancerous cells and the healthy ones. So in 2012, the FDA approved a drug that attacks this specific error in the hedgehog pathway, and my research is focused on making this drug better. I'm trying to create a light responsive version of this drug, meaning as shown in figure 4, it can essentially be taken in its inactive form and then activated by light being shined directly onto the cancerous cells, therefore drastically reducing the harmful side effects of the current drug.
Thank you. Questions for the presenter, please. First question for you. What are barriers?
Barriers. I'm not sure what that's like. Barriers to my synthesis or to my project? Is that the question?
Yeah, Dr. Webb, why don't you clarify that in the chat? I'm sorry. I'll move on to another question. So could this deactivate the hedgehog pathway entirely and lead to no cancerous cell production?
Yeah, so essentially what the drug actually that I'm making acts as is a smoothened inhibitor, which is the error essentially that occurs that causes the cancerous cells. So in the hedgehog pathway, smooth-end is a very important transmembrane protein. But if it is left on, which can happen when something is mutated within the body, then it just continually is regulating these cells, which then produces the cancer cells. So the drug is essentially trying to repress smooth-end so that it's not continually turned on and it's not producing the cancer cells.
Okay. And can you say more about how light is shined on the cancer cells?
Yeah, so this can happen sort of in two ways. Researchers are already working on this. There is this sort of LED tape that has been produced that can go like directly into the body and be put onto the cancerous cells. And then there's also sort of just the aspect of surgery. So like during the time when the tumor or whatever would be removed, then they can also, the surgeons can also shine light directly onto that area to get more of it.
Okay, thank you. And I believe last question for you. What step in your synthesis is the most difficult to accomplish?
Oh, definitely. So I've actually run into some problems with my synthesis because of the positions of the different molecules. And so right now I am on my third attempt. I believe the second step was really the most difficult of it because I had a lot of leftover solvent from the first step, and I had to try to get rid of that by placing it on what's called a high vac, which kind of just evaporates it all. But it took me a very long time to get that all gone and move on to my next step. So it's kind of just the waiting and seeing what's going to happen.
Thank you.
Excellent. Thank you, Samantha. And that concludes all of our poster presentations for today. So excellent job to all of our presenters. Congratulations on your research and on your presentations today. So I do want to just remind you all to please make sure to check out all of the concurrent sessions that are happening this afternoon. They start at 1 p.m. It's a great opportunity to see all of the incredible projects and research happening at Albion College. And thank you all again for joining us today, and have a wonderful rest of your day.
Thank you to all the presenters. Tremendous job.